some stations that are going to be microscopic images off of projected slides. Turn to page 25. Those are two possible questions that would be projected images on slides. Everybody write that down. Page 25, we have pyramidal cells. The cell bodies are cone-shaped, and so they sit down, it looks like a pyramid, so they call that pyramidal cells. And then on the right is Purkinje cells, which I found in the cerebellum. But they have a bunch of dendrites with one axon. Okay, you do need to know, not only to visually recognize those two cells, you also need to know where they're found. Look in the little boxes. Pyramidal cells are found in the cerebrum, the cerebral cortex. And the Purkinje cells are found in the cerebellar cortex. This is why spelling counts in the anatomy lab. Cerebrum and cerebellum are two completely different structures, but they're spelled very similar. You see that? That's why spelling counts. Okay. There'll be more microscopic images next week when we do the census. All right, let's turn to page 22. We will be covering the rest of the brain in lecture next week. But here it is in lab, so we're off by 1 week. The cerebrum comes in two hemispheres. What's in the purple line would be the left cerebral hemisphere. Okay. That whole thing on both sides is a hemisphere. Each hemisphere has 4 lobes. The largest one is the frontal lobe, there in red. The most superior one is the parietal lobe, that's in blue. And the most posterior one is the occipital lobe, that's in green. Do those words sound familiar?
Yes.
They parallel the cranial bones, don't they? Okay.
That's nice.
Now, when this picture was taken, the brain was kind of tilted back a little bit, so you saw most of the frontal lobe and very little of the occipital lobe. So there's a large brain up there on the top of the cards. I took these pictures and I got it a little bit more straight on the superior view, not with it leaning back. So same parallel lines, you can see that a little bit more of the occipital lobe. Okay, it's the same lines.
So the one whole side is the cerebral?
What's that?
The whole back side is just the cerebral?
It's the one hemisphere,
Oh.
specifically the left cerebral hemisphere. But on the right cerebral hemisphere, I'm breaking it up into lobes. But you can break either one up into lobes, and each hemisphere has 4 lobes. Each hemisphere has 4 lobes. From lateral view, we can see all 4 of them. Same red line is frontal, blue line is parietal, green line is occipital. But the one that we couldn't see from the superior view is the temporal lobe there in orange. Everybody got all 4 now? Frontal lobes are the largest, by the way. All right, the yellow arrow is on the cerebellum. So that is how you can see here how 2 words spelled very similarly are 2 completely different structures. The cerebrum, that has the pyramidal cells, is outlined with the 4 lobes. The cerebellum is the second largest structure of the brain. Cerebrum is the largest structure of the brain. Cerebellum is the second largest structure of the brain. That's the yellow arrow. That's what we, actually the last slide today in lecture was the cerebellum, part of the mesencephalon. Same color lines and arrows for the large brain, which is up on top. Everybody got these? All right. Now the cerebellum actually also has two hemispheres, so both of these yellow arrows are on each of the cerebellar hemispheres.
Oh.
The two cerebellar hemispheres are connected by a structure called the vermis. It's the green arrow. Now, just for our sheep brain dissections later, I will warn you. In our brain, the vermis is smaller than the cerebellar hemisphere, so it's sitting in a valley. Everybody see that?
Mm-hmm.
In the sheep brain, the vermis is bigger, so it's actually a ridge that sticks out. Okay? All right,
Do you still just call them left and right cerebellum
cerebellar hemispheres.
hemispheres?
Now, if you cut the vermis in half, mid-sagittal plane through the vermis, you will see the white matter that looks like a tree. That's the arbor vitae, tree of life.
Can't use tree of life on the
No.
one.
Arbor vitae. Okay, so this is the white matter in the vermis, all right? Arbor vitae is the white matter in the vermis. You guys remember the spinal cord? The gray matter is on the inside, the gray butterfly. White matter is on the outside.
Mm-hmm.
Does this ring a bell?
Yes.
Mm-hmm.
In the brain, it flip-flops. In the brain, the gray matter is outside, and the white matter is on the inside. That's why the arbor vitae is the white matter, and it's in the center. But that's also true for the rest of the brain. Gray matter's on the outside, white matter's on the inside. Are we clear? All right, let's go to the brainstem. There's 3 sections of the brainstem. Yellow is mesencephalon. That's right in order than how it is on the left column. Red is pons. Green is medulla oblongata.
Okay.
Everybody got the 3 regions of the brainstem? From top to bottom, mesencephalon, pons, medulla oblongata. On our brains, the pons is a bulge on the brainstem. This is a brainstem model, also has diencephalon. Now you've at least seen the word diencephalon in lecture today, right?
Mm-hmm.
Diencephalon is the thing on top of that. But same color arrows for the brainstem.
So mind.
Everybody got this? All right, now let's look at the superior aspect. We're on the brain, major aspects. Now we're on superior aspect. Yellow arrow is the longitudinal fissure. Remember I drew this on the whiteboard last week? Fissure is the deep crack, sulcus is shallow crack. A ridge or bulge is a gyrus. Okay, so this is a deep crack, and when I was learning the brain as a student, I'd always tie these cracks to what does it divide. And the longitudinal fissure divides the left and right cerebral hemispheres.
Mm.
Everybody see that?
Mm-hmm.
Longitudinal fissure divides the left from the right cerebral hemisphere. Blue arrows are on the central sulcus. Yellow, it's a shallow crack. And the central sulcus goes off to the side. And doesn't make sense, does it? It's central. I'll explain the name later, it actually just makes sense. But for the central sulcus, it divides the frontal lobe from the parietal lobe. Central sulcus is the border between frontal lobe and parietal lobe. Look at this picture with the blue arrows. Kind of burn it into your skull. Everybody got it?
Mm-hmm.
Now look at the right picture. You see where the red line meets the blue line?
Mhm.
That's the line between frontal lobe and parietal lobe. That's the central sulcus. Do you see that?
Mm-hmm.
This line right here, that's the central sulcus. All right. This is the transverse fissure. Green arrow is a transverse fissure, and it separates the cerebrum from the cerebellum. This taking these fissures and sulcuses and naming what it divides helps to understand the brain. And what you understand, you don't have to memorize. Everybody got that one?
Mm-hmm.
Red arrow is on the lateral sulcus. If you look at it carefully, you see it's kind of deeper than the other ones, isn't it?
Yeah.
And that's why some textbooks and some websites, you might see it listed as lateral fissure. It's not wrong. It's both called lateral sulcus and lateral fissure because it is deeper. Okay? And the lateral sulcus separates the temporal lobe from the frontal and parietal lobes. Blue arrow is again the same central sulcus.
What's blue?
What's that?
What's the
Blue, central sulcus, just like on the previous slide. All right, in front of the central sulcus, or before it, is the pre-central gyrus. It's a ridge that sticks out.
Pre-central gyrus.
Okay. Everybody got this? Now, if you want to contract any muscles on your right side, action potentials start or initiated in that green area. Contracting skeletal muscle is somatic motor. Everybody got it? This is what's referred to as the primary motor cortex. And because everything in your body, both sensory and motor, goes through decussation, what does that mean? From this morning.
Oh.
Decussation. Crossing left to right.
Oh.
You have to initiate things on the left cerebral hemisphere in this green area to contract muscles on the right side. Everything crosses left and right. If you are touched on the right side, since this is the left cerebral hemisphere, those action potentials end up in the orange area called the post-central gyrus. But it crosses left to right, so if you're touched on the right side, the action potentials end there in the orange area. That would be somatic sensory. Everybody got it? Are you tracking with me? Or is the content coming in too efficiently for you? Should I say a few ohms to dilute it?
No.
Okay. Thus, these two areas in the dashed lines are the somatic area of the brain. Somatic motor in green, somatic sensory in orange. The central sulcus runs down the center of the somatic area of the brain. The name actually makes sense.
Mm-hmm.
And remember, because this is the borderline between frontal lobe and parietal lobe, the precentral gyrus is the most posterior part of the frontal lobe, and the postcentral gyrus is the most anterior part of the parietal lobe. Does this make sense?
Mhm.
Okay. All right, this is sheep brain. This is showing you some dura mater. The dura mater is like a thin sheet of leather. It's pretty thick. It's tough, but it's actually just physically thin. It's just a thin leather-like layer, dura mater. What's actually stuck on the brain there, the orange arrow, is pia mater. The arachnoid is completely delicate, and it's stuck on the inside of the dura mater, and as soon as the butcher houses tear out the brain, pretty much it falls apart. It's so delicate. So I will not be pinning any arachnoid.
Cool.
See, you guys are becoming better students already. I only have to say that once now. I said that once, all of a sudden I said, you guys are actually present. First couple weeks I'll have, I say like 4 times. You guys are getting there. All right. So this is a model up here on the top of the cart. And it shows dura mater, but only on the midline. That's the blue arrows. And on the right side, it actually shows pia mater still stuck on the brain. Now look where it says dura mater. Do you see where it says falx cerebri? That is not a synonym. That's not a synonym. Let's show you what falx cerebri actually is. Here's that same model from the right side, view from the right side. I can actually pull out the dura mater. Now it looks like that.
Whoa.
Everybody got it? So this same blue arrows here are the same blue arrows here. This is looking from the top. That other thing is looking from this side. But it's looking at the same blue, dark blue arrow. Everybody got it? The falx cerebri is the specific sheet of dura mater that sits in the longitudinal fissure. And that's the light blue arrow. It is the specific sheet, vertical sheet of dura mater that sits in the longitudinal fissure.
So if you pin it there, we have to call it the falx.
Falx cerebri.
And if you pin the top, we call it the dura mater.
Dura mater.
Cool.
Because all falx cerebri is dura mater, not all dura mater is falx cerebri. Does that make sense?
Yes.
Okay? Any questions?
Yeah.
In lecture, we'll learn that there's a sheet of dura mater in the transverse fissure called tentorium cerebri, and it's horizontal. So between the falx cerebri and that tentorium, it's like a 90-degree angle. One is horizontal, one vertical. But that's the only one you have in lab, falx cerebri. Just make sure you know that they're not synonyms. Falx cerebri is the specific vertical sheet of dura mater in the longitudinal fissure.
And does the pia mater go over the entire, both sides, the whole? Okay.
And in all the sulci and all the fissures, it is hugging stuff on the brain. There's actually no brain that is uncovered.
So the cerebellum and the cerebrum.
All of it.
Okay.
In fact, all 3 layers are on all of it. In the brain, we'll learn this in lecture, but in the brain, for the brain, there's actually 2 sheets of dura mater, not just 1. All right, so there's a longitudinal fissure here in the sheep brain. So when we do sheep dissections, we might do it in groups of 3 because we got 30 in here. Congratulations, you guys. This is week 8. I've never had an anatomy lab with 30 students in week 8. I mean, not even close. Usually by week 8, it's down to about the mid-20s at best. I've had some semesters where it's down to 18 already,
Oh my God.
and you guys are at 30. So let's do our dissections today in groups of 3. So we'll have 10 groups of 3. How's that sound? A third of you are going to do external structures. That would be like this on the screen. A third of you are going to do mid-sagittal slicing, and then a third of you are going to do this coronal section. So I'll show you what it looks like on the slides when we get there. And it's a good thing that we have slides on the PowerPoint, because there will be 2 sheep brain stations on lab exam 3. So while we're doing the dissections, just be sure to remember that you're not just fooling around this afternoon. You just actually want to be serious about the dissections, because there'll be dissected brains. But there'll be a third of you doing external, and those that do external, once I'm going to split you up, since you have 3 different types of dissections: external, sagittal, coronal. In each groups of 3, I'm going to split you up in groups of 3. So once you've been checked out in those lists of structures, you've got to show the other 2 groups the external structures. Sagittal's got to show the other 2 groups of sagittal structures. Coronal's got to show the other 2 groups of coronal structures. Everybody got it? You're actually going to show each other. So you've got to be checked out and know what you're doing, so that you don't lead other students astray. Everybody got it?
Yep.
Okay. All right, so external, this is longitudinal fissure with the 2 cerebral hemispheres in purple. Here you can see in the cerebellum, the smaller cerebellar hemispheres off to the side, and notice how the vermis is a ridge instead of a valley. Green arrow is a vermis.
Okay.
And it's bigger in the sheep brain, smaller in our brains. In our brains, it's a little valley. Sheep brains, it's a ridge. It's a little tag of dura mater off on the side. I don't think any of the sheep brains that we have dura mater. Don't worry about it. Dura mater is really easy. It's just this thin leather sheet covering it. But on this photo, you can see this kind of whitish snot stuff on here. That's pia mater.
Okay.
Actually stuck on the brain. What's that? Cerebral hemispheres. And you can see the longitudinal fissure dividing the 2 cerebral hemispheres. Any other questions?
Oh, okay.
Any other questions?
What's up, blue arrow?
Which one?
The blue?
Blue is dura mater. Just a little tag of it.
Wait.
Any other questions? Now we're to upper sagittal aspect, so still left column, but we're getting close to the bottom. Orange arrow number 1 there is the corpus callosum. Corpus callosum is white matter. It has commissural fibers connecting the left and right cerebral hemispheres. You guys remember what commissural fibers are from lecture? Is that too
No.
long ago? Don't remember this morning?
Is this part of your drawing?
I wrote it on the whiteboard.
Oh.
Commissural fibers and with decussation. Oh, that's right, those are together. That's probably why you don't remember.
I'm gonna do excellent.
But it's crossing left and right. It's fibers that are crossing left and right. And that's why in the mid-sagittal section, you see this clear as day, because it's connecting right and left cerebral hemispheres, crossing left and right.
Okay.
Underneath it is the septum pellucidum. Okay, this is
22.
mid-sagittal, exactly right in the midline, correct?
Uh-huh.
I'm going to explain this, and hopefully you'll do better than Tuesday lab.
Okay.
Because, okay, in all fairness, Tuesday lab just took the lecture exam, and I had to try to explain this. And it was like explaining how to build a pyramid. All right, here we go. Are you guys ready?
Mm-hmm.
On the mid-sagittal plane, what things do you see on the mid-sagittal plane that are lateral?
The same? The exact same?
On the mid-sagittal plane, what lateral things do you see? It's not a trick question. Nothing. Nothing. Lateral means you're off the mid-sagittal plane.
Oh, okay.
All right, so then why here, down there in the left column, under the upper sagittal aspect, are they listing lateral ventricles? You cannot see lateral ventricles on the mid-sagittal plane. Are you following me now? Okay, this right here is actually a model taken from a human brain. Remember I told you that the ventricles and aqueducts are the remnants of the hollow space in the hollow neural tube? And so I drew that thing with the CSF on the whiteboard today. Well, what they did was they took hot latex, shoved this needle up into the central canal of the spinal cord, and injected hot latex into the brain, into the ventricles and the aqueducts, and this is what came out.
Oh my.
Is that wild? This used to be just a slim little channel in the neural tube, but when the brain got big, this is what it became. But in order to do that, they had to inject this hot latex with a lot of pressure. And when it does it with a lot of pressure, the lateral ventricles, which are the two big ram's horns, do you guys see that even back there? The two big ram's horns, artificially separated. And actually right here, now look carefully where my finger is, in your brains, the two ram's horns actually touch. But when they made this, there was so much pressure with that latex, it pushed them artificially apart. That's not how it is in your brain. Right here, they actually touch. Are you following me?
Mm-hmm.
And where they touch is on the mid-sagittal plane.
Mm.
That is the septum pellucidum. What does it say there in the left column under septum pellucidum?
Curtains between lateral ventricles.
Does it make sense now?
Curtain?
That's the only thing of the lateral ventricles that you can see on the mid-sagittal plane is the septum pellucidum where these two babies touch. Does that make sense now?
Yes.
Good, hope so. Because you guys don't have an excuse of having a lecture. Tuesday lab, they're just like, you know, like how puppy dogs do. Okay. Blue arrow on the number 4, that is thalamus. The thalamus kind of looks like a football, actually more specifically a rugby ball. You know, football's kind of oblong. Well, football actually has points. These don't have points, but if you look at the shading of that number 4, can you see how it's kind of like an oval round ball?
Where does it end? Can you trace it?
Underneath there?
Can you trace the whole thing?
Oh my gosh. Okay. Behind here a little bit.
Okay. Okay, yeah, I'm glad you traced that.
Okay. Can you see how it's kind of an oblong ball?
I thought it ended where the pink, do you see that
No.
pink line?
That's choroid plexus of the third ventricle. Oh, never mind. Okay. This whole thing where the number 4 is. Like a football. Well, this is on just the left side, right? This is the left half of the brain. That means you have 2 thalami, one on the left and one on the right.
So thalami.
Got it?
With an I?
Yeah, for plural.
No S-I. M-U-I.
Well, you're going to only say thalamus because I can only pin one.
Oh, I thought you were trying to say like, call it a thalami, because there's 2. You're not
Just
saying
as
that.
You take off the US and put an I there, and that's what everybody has. You have 2 footballs in your brain. One on the
Okay.
left, one on the right.
Thalamus.
Everybody got it?
Thalami.
90% of you. So 30, that'd be 27 of you. Us actually will connect the left one to the right one, and that's the green arrow, interthalamic adhesion.
Interthalamic.
You can also call it intermediate mass. Those are synonyms. Full
Ugh.
point for either.
That's way better.
3 of you do not have that at all. The thalami are disconnected. Okay?
What? three of us lost it?
Yeah, only 90% of people have the interthalamic adhesion, 10% do not.
Oh, okay.
What's that?
I thought he knew something about
It's,
that.
it's genetics,
Does it cause a difference?
yes. You have very difficulty with multiple choice questions.
Well, mine is gone then. It's fused to the lateral part of my brain.
There is no known defect for not having. All right. Everybody started wondering. Outlined here in red is the outline of the third ventricle. Basically, the medial wall of the thalamus and hypothalamus form it on both sides. So that red dashed line is the outline of the third ventricle.
So how are we gonna know if you're pinning that versus pinning the football versus pinning the intermediate mass?
Great question.
Oh my
So
god.
if you look at, oh, you want me to enter it?
Yes!
Oh. I'll show you why in just a second.
Okay.
Now look at that outline. Doesn't that look like a duck head?
Uh-huh. Well,
And like,
oh yeah, I see it.
you see it kind of like a duck head?
Mm-hmm.
And it's like that interthalamic adhesion is like a perfect eye for the duck. Let's see if this thing will work here.
See, you gotta look, the beak is that way, not that way. Yeah. You don't see that?
Remember, this is a model of the ventricles.
Looks more
Do
like
you kind of see a duck head
Yeah.
with an eye?
Yeah.
kinda looks more like
You
a
guys
dinosaur.
connecting the dots?
Yeah.
This is what I would pin for the third ventricle.
Cool.
Because you wouldn't know if I pinned this over here whether I was pinning the thalamus or not.
So the latex duck.
Yeah, the duck head.
Latex duck.
They call it third
head.
ventricle, don't call it duck head.
Okay. Third.
But you can see when they injected this brain with the hot latex, this brain was one of the 90%ers. Because it has an eye. Everybody got it?
Mm-hmm.
That hole was because this brain had an interthalamic adhesion.
So is
Everybody
it
catching what I'm saying?
is it actually a space? Like
Well,
it's an
It's
open?
because something is solid. This actually all was a space filled with cerebral spinal fluid.
Oh, okay, I get it. Okay,
That eye was something solid, the interthalamic adhesion.
I got it. Okay.
Okay? Does that help?
Yeah.
All right, let's go. Now we're in upper coronal aspect. We're at the very bottom of the left column. Blue arrow is just cerebral cortex. Red arrow is the caudate nucleus. The caudate nucleus is a ring of gray matter around the thalamus.
Caudate nucleus. A ring
And
of
remember, the thalamus is football shaped. So do you kind of see this thing right here? It's a little bit bigger because this is the structure surrounding the thalamus, but can you kind of make out a football shape here? And on this model, the manufacturer made the gray matter not gray, but kind of pink. But you can see the gray matter is on the outside, and everything on the inside is white matter. Gray matter and white matter switch in the brain from the spinal cord. In the brain, the gray matter is on the outside, white matter is on the inside. Everybody got it? All right, this is supposed to be gray. And if it was gray, what is specifically causing this out here to be gray?
cell bodies.
Let's be specific. Turn to page 25.
Oh boy.
What is causing this to be gray?
Oh, the perimordial cells or
The cell bodies of the pyramidal cells. Because pyramidal cells are in the cerebral cortex. Blue arrow. Everybody got it? Everybody know where we find pyramidal cells?
In the
Gray matter of the cerebral cortex.
in the gray matter. Okay.
Same color arrows here. Can you kind of make out the football shape?
Yeah.
A ring of gray matter around the football, around the thalamus, caudate nucleus. Red arrow.
Caudate nucleus. Oh.
Okay, I think we're done with the left column. You ready to move on?
Okay.
All right, now we're on the top of the right column, and it says lower sagittal aspect, and you might look at this and you think, oh, that's not sagittal. I know it's not. But the pituitary gland actually is mid-sagittal. It's just that when your manufacturer makes this brain that you can divide in half, they got to pick a side to put the pituitary gland. So you can see that they picked a side. But actually, this pituitary gland is in the direct midline. It's mid-sagittal. Okay?
And
Pituitary gland.
can we call it the hypothesis?
You can call it the hypothesis. In fact, that'll make more sense when I do endocrine.
Okay.
Do we have to say gland?
Pituitary gland, yes. You either say pituitary gland or hypothesis.
Hypothesis gland?
No,
Oh, okay.
you don't have to say hypothesis.
Gonna go with that one then.
The pituitary gland was there first, and then the sphenoid bone formed around it. Do you know what formed around the pituitary gland?
The sella tertia.
Sala turcica. Pituitary gland is first, sphenoid bone formed around it. Now you have a sala turcica. All right, now let's do the sagittal. The blue arrow here, the number 8, is hypothalamus. You can see that it is below the thalamus, number 4. Hypo means below.
Okay.
Here, listen to this now, because this is going to help you people if you're doing the sagittal section on the sheep brain. This is how you find it. That pituitary from the previous slide, the orange arrow thing, that little punching bag looking thing, the manufacturers put a little stick on it, and they stick the stick right here. Because you have to look carefully, it looks like a little hole. That's where the little pin goes in. That's where the pituitary comes out of the brain. Everybody got it? All you have to do is find where the pituitary stalk is, where the pituitary comes out, and the thalamus, draw a straight line, and the hypothalamus is right in the middle of it.
Mm.
Go from pituitary stalk to thalamus, right in the middle, hypothalamus.
Okay.
And it'll be below the thalamus.
Below.
In lecture we'll learn why, because the hypothalamus controls the pituitary. That's why it's just above it. Do I need to repeat that on how to find the hypothalamus? Everybody got it? And so that's what the orange arrow is. It's showing you where the pituitary comes out.
Okay.
All right. The entire yellow dashed line is the mesencephalon.
Okay.
On the anterior side of the mesencephalon are ascending and descending, that's vertical lines, ascending and descending tracts we call cerebral peduncles, so that's the yellow arrow. Anterior mesencephalon, vertical lines, cerebral peduncles.
Cerebral peduncle.
Anterior mesencephalon, vertical lines, cerebral peduncles.
Cerebral peduncle. 31.
Anterior mesencephalon. What's the mesencephalon? That is the mesencephalon. To the right is anterior. Anterior mesencephalon, vertical lines, cerebral peduncles.
Are they pretty obvious?
Uh-huh.
Okay.
You're going to see it a little later.
Because they're not so obvious here.
red arrow is the cerebral aqueduct. Since it's going through the mesencephalon, you can also call it the mesencephalic aqueduct.
Mesencephalic aqueduct. Okay. And
What
that's
I would write next to cerebral aqueduct is that it is a pipe
a pipe.
that connects third ventricle to fourth ventricle. And the fourth ventricle is this purple arrow down here.
Okay.
The cerebral aqueduct is a pipe that connects the third ventricle, remember the duck head?
Mm-hmm.
Connects the third ventricle to the fourth ventricle, purple arrow.
So if you pin it anywhere above that purple line, it's still the pipe.
Yeah, if I pin cerebral aqueduct, it's going to be right smack in the middle of it. I'm not going to get close to one of the ventricles,
Okay.
because then you'd have probably a legitimate reason to bitch out.
Mm-hmm.
Right smack dab in the middle if I do cerebral aqueduct. Okay, on the back side of the brain, and the brain stem are 5 bumps. The most superior bump is this, that's the pineal gland or pineal body. You can say either pineal gland or pineal body.
Pineal body.
That's the light green arrow.
34. That was the fourth ventricle. Yeah.
Then underneath the pineal body are 4 bumps. And the 4 bumps are called corpora quadrigemina.
Corpora quadrigemina.
How come I only see 2 here?
Because it's half of the brain.
Because this is the left 2. There's 2 on the right. That's the 4 bodies, corpora quadrigemina. But it is listed there on the top right of page 22. You do also have to know this. The 2 upper bumps, and here it's only on the left side, but the upper bump is the superior colliculus, and the lower bump is the inferior colliculus.
So do we need to say the superior colliculus of the corporal quadrigemina?
No, because I don't think any of the lab exam form lines are long enough. Just
Okay.
say superior colliculus.
And we're good.
So you would not pin the corpora.
I could, especially on the intact brainstem that does show all 4.
So if it's all
And I would
four,
put the
that's-
pin right in the middle, right in the middle of all 4, and I would say, what are these 4 bumps?
So you're only gonna ask that if it's 4 of them.
Or I can cut a piece of string and then circle the 4 bumps and have the pin on the string.
But it's only if there's 4 of them.
Yes, you gotta see 4 for quadrigemina.
Okay.
If it's 1 bump, it's a colliculus.
Okay, one bump.
That's a good question. Hey, this is the reason why you should ask questions. You can get a lot of answers if you ask questions.
Yeah.
You have to have all 4 to be corpora quadrigemina.
Mm-hmm.
If I'm pinning 1 bump, it's a colliculus.
Superior or inferior colliculi. Or colliculi, right? Because you only
Yep.
pin 1?
Colliculi is plural, and you have 2 superior colliculi. But if I'm pinning 1, I can only pin 1, and that would be superior colliculus.
Okay, colliculus is singular.
Yeah.
Okay.
Everybody got it? So if I pin this, this would be superior colliculus.
And
This
inferior.
would be inferior colliculus.
Okay.
But
So,
on the full brain, you have a pair of each.
so can we
colliculi.
technically cross out the colliculi?
Yes, I would.
Oy.
Okay, so here's the brainstem. Even though this is mesencephalon, since this is on the anterior side, and can you see the vertical lines?
Yeah.
That's cerebral peduncles. This is pons. This is medulla oblongata. This is the anterior side. So on the posterior side, you can see the 5 bumps. Everybody see the 5 green arrows?
Yes.
Pineal body on top. Here's the two superior colliculi. Here's the two inferior colliculi. 5 bumps on the back side of the brainstem.
So where's the inferior?
So this side over here is the posterior.
Okay, okay.
The yellow?
Yellow is cerebral peduncles.
Cerebral peduncles. Okay.
You could say mesencephalon, and it would be right, but it wouldn't be as specific as cerebral peduncles. So mesencephalon would get you a half point. Oh, by the way, we didn't really have much of this on the last lab exam, and we probably won't. We had a few stations that had it on the last lab exam. Probably won't have any on the next one. But whenever there is a parentheses on the card, on the question card, on the desk, and it says be specific, this is a good example, by the way, of asking that. If I pin this, and it says in parentheses, be specific, that's why mesencephalon only gets you a half point. This is the cerebral peduncles of the mesencephalon. Cerebral peduncles for the full one, because it said be specific. Does that make sense?
Uh-huh.
Hopefully you guys I know some of you catching the clue, but hopefully more of you will catch a clue that a lot of the questions are giving you hints to the answer. Like be specific. Oh, this is pinning something where there's 2 possible answers and 1 specific. Yes? Mesencephalon now. Just write cerebral peduncles. Okay.
Okay. Cerebral peduncles.
All right, now we're going to be on the inferior aspect. We're all done with the lower sagittal aspects. Let's just do a few of these and we'll stop for break. This is olfactory bulb on top, olfactory tract below it.
Olfactory bulb and tract.
Even though it says cranial nerve number 1, technically this is second order neurons. But we will call this cranial nerve number 1. You guys remember ethmoid bone, cribriform plate? These 2 guys are sitting on top of the cribriform plate.
Oh. Oh, and
remember
that's
how the
why the 2
cribriform
sides.
plate has
Yeah.
like 20 holes on each side?
Yeah.
The neurons going through the holes is actually cranial nerve 1. These are second order neurons, but just for our purposes for lab exam 3, you can say that this is cranial nerve number 1.
Okay.
All right. I'm gonna do this here and then we're going to take a break, okay? The blue arrows right here have to do with the optic nerve. So here's 2 eyeballs with the cornea. Okay? And this is like superior view. Again, as a student, I would do a lot of drawings just like this. All right. The optic nerve, and this is cranial nerve number 2. Actually comes out of the eyeball. So the optic nerve only has axons from 1 eyeball. So far, so good? That is actually the dark blue arrow right here. If you think about it in terms of being an X, these are the top arms of the X. Everybody see this X right here? The top arms of the X going to the eyeballs is cranial nerve 2 optic nerve. The middle of the X, some of the axons are crossing left to right. What's the word for crossing left to right?
Something with a D.
Decussation. But right in the middle of the X the optic chiasm. It's on the midline.
Optic chiasm.
So this lighter blue arrow that's going to the midline is on the optic chiasm. Then the lower arms of the X that enter the brain, that's the optic tract. And that's this other light blue arrow over here. And it's kind of in the shadow, so it's hard to see, but this actually enters the brain. And this red dot that I drew right here, this is the pituitary stalk that the pituitary gland dangles from. It's not on page 22, don't bother looking for it. But it's going to help you with the brain dissections, the sheep brain dissections later.
Is there any view of something that we're gonna see where it looks like that? Like an X and
Uh-huh.
there I see.
Okay, let's go ahead and take a break. Short one, be fast. If you have to go to the bathroom, hopefully you have to bring in the newspaper.